Welcome to this lecture on human physiology, specifically focusing on endocrine glands and hormones. Today, we are going to explore how your body communicates with itself over long distances. We will uncover the biological wireless network that keeps you alive, regulates your growth, and maintains your internal balance. By the end of this session, you will not only understand the biology, but also the mathematics behind how these chemical signals work. To begin, imagine you are the principal of a massive school with trillions of students, representing the cells in your body. If you need to send a specific message to just the science teachers, you would not run to every single classroom. Instead, you would broadcast a message over the school radio system. Only the teachers with the right radio receivers would hear and act on it. In your body, the endocrine system works exactly like this wireless broadcasting network. It allows different parts of your body to communicate and coordinate complex tasks without needing direct physical connections, like nerves. Now that we understand the broadcasting analogy, let us look at the broadcasting stations themselves. These are the endocrine glands. They are specialized, ductless organs that synthesize and secrete chemical messengers directly into the bloodstream. The word ductless is crucial here. It means they do not have tubes to carry their secretions to a specific surface, unlike your sweat or salivary glands. Instead, the chemical messengers they release, which we call hormones, travel freely throughout your entire circulatory system, reaching almost every corner of your body. Since these hormones are flowing everywhere in the blood, you might wonder how they know where to go. The truth is, they do not. However, hormones only affect specific target cells that possess the exact matching protein receptors for that specific hormone. Think of it as a lock and key mechanism. Once the hormone finds its match and binds to its receptor, it triggers a cascade of cellular events. This binding alters the function of the cell, such as changing its metabolism, activating specific genes, or stimulating cellular growth. Understanding how these target cells react brings us to the ultimate purpose of this entire system, which is maintaining homeostasis. Homeostasis is the stable internal environment your body absolutely needs to survive. The endocrine system regulates long-term, ongoing processes, such as your overall physical growth, your daily metabolic rate, your sleep cycles, and your reproductive functions. Without this system, your cells would be completely isolated. They would be unable to coordinate necessary responses to everyday changes, like digesting a heavy meal or reacting to sudden stress. To truly appreciate this delicate balance, we must look at what happens when the system fails. In the real world, we see the critical importance of the endocrine system during such failures. For example, consider the pancreas, which produces the hormone insulin. If the pancreas fails to produce enough insulin, the person develops a condition known as diabetes mellitus. Because the insulin signal is missing, the target cells do not know they should absorb sugar, and the patient can no longer regulate their blood sugar levels effectively. Knowing what happens when hormones are missing, let us now explore how we measure their activity when they are present. To understand how hormones actually trigger a response, we must look at how they bind to their receptors using the principles of chemical kinetics. Biology is not just qualitative, it is highly quantitative. By looking at the relationship between the hormone concentration and the amount of bound receptors, we can predict exactly how strong a physiological response will be based on how much hormone is present in the blood. This quantitative relationship leads us directly to the fractional occupancy formula, which you see here. Conceptually, this formula tells us what fraction of the available receptors are currently activated by a hormone. Let us break down the symbols. Y represents the fractional occupancy, which is the percentage of total receptors bound to a hormone. H, in brackets, represents the concentration of the free hormone in the blood, measured in moles per liter. Finally, Kd is the equilibrium dissociation constant. This constant represents the exact hormone concentration at which half of the receptors are bound. When the hormone concentration increases, the fractional occupancy Y also increases, leading to a stronger physiological response. Conversely, a high Kd means the hormone has a low affinity for the receptor, requiring much more hormone to achieve the same effect. Now that we have our formula, let us apply it to a real biological scenario involving insulin. Suppose a patient has just eaten and their pancreas secretes insulin, resulting in a blood concentration of 4 × 10⁹ molar. We are given that the dissociation constant for the liver receptors is 1 × 10⁹ molar. We want to find out how strongly the liver is being stimulated. We start by substituting our known values into the fractional occupancy formula. Next, we add the terms in the denominator together. The units and powers of 10 cancel out perfectly, leaving us with 4 divided by 5. This gives a final answer of 0.80. Physically, this means 80% of the insulin receptors on the liver are bound, indicating a very strong signal to absorb sugar from the blood. Let us look at another example, this time working backward to find the required medication dosage for a patient with hypothyroidism. Here, the doctor knows the target physiological response requires 90% of the thyroid receptors to be occupied, so Y is 0.90. The receptor's dissociation constant is 2 × 10⁻⁻⁻ molar. We need to find the required hormone concentration. We plug our known values into the formula. To solve for the hormone concentration, we first multiply both sides by the denominator to remove the fraction. Next, we distribute the 0.90 into the parentheses. Then we subtract 0.90 times the hormone concentration from both sides to group our unknown variables. Finally, dividing by 0.10 gives us 1.8 × 10⁻⁹ molar. This tells the doctor the exact concentration of medication needed in the bloodstream to maintain a healthy metabolism. Having calculated these values algebraically, let us now visualize this relationship on a graph. What you are looking at is the standard hormone receptor binding curve. The horizontal axis represents the hormone concentration in the blood, plotted on a logarithmic scale. The vertical axis represents the fractional occupancy, ranging from 0 to 100%. Notice the smooth S shape of the curve. At very low concentrations, almost no receptors are bound. As concentration increases, the occupancy shoots up rapidly before eventually leveling off as the receptors become saturated. Pay special attention to the dashed line. It shows exactly where the fractional occupancy is 0.5. If you follow this down to the horizontal axis, it points directly to the dissociation constant. This diagram beautifully summarizes the chemical kinetics we just calculated. We have covered a lot of ground today, so let us summarize the most important ideas. First, the endocrine system is your body's ultimate chemical communication network, utilizing hormones to broadcast vital instructions through the bloodstream. Second, these hormones do not affect everything. They act like a lock and key, binding only to target cells equipped with specific receptors. Finally, By understanding the mathematical relationship between hormone concentration and receptor binding, we can precisely predict physiological responses. This mathematical foundation is what allows modern endocrinology to treat complex hormonal disorders effectively. You now have a solid understanding of both the biology and the math behind endocrine function.